So welcome to this webinar again. My name is Elisabeth Gasteiger, and we will explore together what is needed to make meaningful queries into UniProt, what kind of data UniProt provides, and how biocurators work. Today with a special focus on plant proteins. This presentation was prepared in close collaboration with my colleagues from the Swiss-Prot groups who are here with me, Emmanuel, who is a plant biocurator, and Marie-Claude, who has also been working as a biocurator and is our outreach manager. The outline for the talk today is as follows. We'll provide first a concise introduction to the UniProt knowledge base. Then we will explore the differences between the two sections of UniProtKB, which are the reviewed Swiss-Prot and the unreviewed TrEMBL, in terms of content and information sources, and I will give you some important background to help you understand how to query and interpret your query results. Then we'll direct our focus to the content of UniProt records and briefly talk about proteomes. And in the last part, we will see in more detail some steps of the biocuration process. And finally, we'll explore together an example of how the biosynthesis of a few natural products is annotated. We will illustrate the different parts with examples of simple and advanced queries. We have decided to focus this presentation on plant enzymes and natural products. Why is that? As you probably know, enzymes are involved in almost every biochemical process that occurs in living cells. And as expected, their genes constitute a large fraction of most genomes. In Arabidopsis thaliana, for example, it's about a third. Natural products, of course, are of major interest in the biomedical field. And the main questions for today will be what types of plant enzyme-related information, natural product-related data can be found in UniProt, and what do you need to know to conduct meaningful queries into UniProtKB. Let's first start with a brief overview on UniProtKB. If you go to the UniProt website, you have access to several databases, and today we will focus on the UniProt knowledge base, or UniProtKB. What is UniProt? It is a database that is freely accessible and provides a comprehensive and high-quality set of protein sequences associated with biological information. The current release, which is 2025-02, there are about 250 million protein records from about 1.3 million different species. UniProt is a collaboration between the SIB in Switzerland, the European Bioinformatics Institute in the UK, and PIR in the United States. Now, during this webinar, we will show you how to search UniProt. Most of you are probably familiar with the simple search box, but we will mainly show today how you can build queries using the advanced search options, which allows to choose specific data fields within which we want to search for our terms, instead of just doing a full text search. Even more complex queries can then be done using a technology called Sparkle. And to make good use of all these query engines, you need to know first what UniProt contains and how the data is structured. And this is what we're going to see today. There are about 18 million green plant entries. To obtain this answer from UniProt, we're going to use the taxonomic term Viridiplantae, or rather its associated taxonomic identifier, which is 33090, with the advanced search interface, which provides suggestions depending on our input. So you don't have to know this taxonomy identifier. This number of 80 million is what we can obtain from the current version of the database, but is of course not, it doesn't fully represent the complexity of nature. Each time you will see this icon, the yellow one with the database and the looking glass. During this presentation, we will provide the syntax of the query with a link to the results on the website. Now we want to see which plant species these 18 million proteins come from. More than 227,000 different green plant species are present in UniProt, in the taxonomy. And among those, about 130,000 have entries in the UniProt knowledge base. We can obtain this number by using advanced search. We can select the taxonomy space to the left side of the query box and then fill in the form. Our next focus will be on exploring the differences between the two sections of UniProtKB, the reviewed Swiss-Prot and unreviewed TrEMBL, in terms of content and information sources, and the implications on you as our users. Swiss-Prot, the reviewed section of the UniProt knowledge base, is reviewed, is expert curated, and contains less than 1% of all the records. TrEMBL, however, that's the unreviewed section, contains more than 99% of the records. It's very important to know, though, that although being tiny in numbers, this small set of expertly curated proteins is extremely crucial for researchers and is also used to improve the huge amount of unreviewed entries in TrEMBL, using automatic annotation and machine learning techniques. Here's an overview of the major differences between the two sections. Swiss-Prot, as I said, is reviewed by expert biocurators. The sequences are reviewed. In TrEMBL, the sequences are not at all reviewed. In Swiss-Prot, there is no redundancy. There's one record per gene, while in TrEMBL, there is some redundancy. There is one protein sequence per record, and there can be many sequences for the same gene. As far as biological information is concerned, in Swiss-Prot, it is extracted from the literature, but additional information can also be propagated from orthologous species or based on computational analysis, which is, however, always evaluated by the curators, while in TrEMBL, the little biological information there is provided by automated systems. To give you a first impression of these differences, we query UniProtKB with a gene name. For example, here the AAE1 plant gene. We find entries from various species. Some of them are reviewed, 2 of them are reviewed, the remainder are unreviewed. And you will note that there is some redundancy. For example, there are 3 sequences for this protein or for this gene for maize. 3 separate protein records. This table here represents 3 different queries in UniProtKB with the number of results broken down into the 2 sections, Swiss-Prot and TrEMBL. For the query, how many plant enzymes are there in UniProtKB? There are about 16,000 entries found in UniProt, which have been manually reviewed, and about 2.4 million in TrEMBL, where the annotations, as I said, are generated automatically. Now let's have a look at the content of a record. This is going to be a very brief overview of the content, and we just want to introduce a few basic concepts in the following slides. As an example, we will examine a UniProtKB Swiss-Prot entry corresponding to an enzyme involved in the biosynthetic pathway of cannabinoid-associated metabolites in cannabis sativa. Each record has a unique accession number here at the top of the entry. And we strongly recommend that you always use these accession numbers when referring to or citing a record, because these are the ones that are unique and stable. You can browse through any record on the website by navigating through the section titles listed on the left. Let's have a look at the content of this record, focusing first on the protein sequence. What defines the protein record is, in the first place, is the protein sequence itself. Every record contains one sequence, but if there are alternative isoforms for the same gene, this is not so frequent in plants. There can be more than one sequence, and in that case, the protein sequence shown by default is called the canonical protein sequence. It's important to note that 99% of the protein sequences in UniProtKB are derived from the translation of nucleotide sequences, either from mRNA or DNA. And that in Swiss-Prot, sequences are verified by biocurators, and if there's a genome sequence available, they are matched and mapped to the genome. In the query we did just a few slides before, we can notice the variation in protein length associated with this gene AAE1. This is primarily due to the fact that the various authors who submitted their data to the nucleotide sequence databases use different gene prediction tools. Remember, protein sequences in TrEMBL are not manually reviewed. You may also notice the different protein names here. And the different lengths and names may make you wonder if these proteins really all have the same biological functions. And in fact, for this particular gene, which is called AAE, acyl activating enzyme, this is quite a generic name, and the reviewed entries for Arabidopsis and cannabis already have quite different sequences and different function, hence also different names. And as far as TrEMBL is concerned, the unreviewed entries, it seems that the automatic annotation procedures have chosen a much more generic name. As I've just hinted at, predicted gene models are sometimes inaccurate and require manual curation. This is a task carried out by our biocurators. This example of an Arabidopsis gene illustrates that the predicted boundaries of exons 2 and 3 here vary quite a lot depending on the different gene prediction tools used. And this is where the work comes in that our biocurators do. Now, after the sequence, let's focus on the biological knowledge presented in an entry. Before showing any details, let me start by saying that all annotations that are added are linked to their sources. The website has these textual labels, which are clickable for details. And behind this, in the original data formats, we use a set of codes from the evidence and conclusion ontology, ECO, which allows you to make very specific queries into the source and potentially also the trustworthiness of an annotation. In Swiss-Prot, the color coding is usually follows the one here with golden flags for the reviewed evidence and gray ones for the automatic annotations. Biological information is spread across the different sections of the entry, and we'll start with the function section. In this entry, a free text description outlines the protein's function, while the catalytic activity subsection offers a more structured description of the enzymatic reaction. The source, as I showed you, is clearly indicated. There is this evidence tag here with a link to the publication if you click on it, because the information has been extracted from the peer-reviewed scientific literature. We'll see later in the presentation how the biocurators do this. In the catalytic activity section, you can see a number of identifiers, which I'll explain right away. All the enzymatic reactions in UniProt are imported from the RIA database, which is an expert-curated knowledge base of chemical and transport reactions of biological interest. We also use information from the so-called EC numbers, which you may be familiar with. That is a numerical classification system at 4 levels for enzymes based on the chemical reactions they catalyze. This classification is established by a committee called the Enzyme Commission. RIA uses the chemical dictionary KEBI to describe reaction participants at pH 7.3. The fact that we use RIA and KB in UniProt significantly helps standardize the annotation of enzymatic reactions and small molecules, and that allows us to describe these reactions more precisely and more consistently, and will also facilitate queries into this data, of course. Another way of representing biological information is used for important sites that can be associated with a specific position in the protein sequence, such as active sites, binding sites, or post-translational modifications. This type of information is often provided using a controlled vocabulary, which is in this case KB again. We move on in our record with the data related to subcellular location. Again, the source is clearly indicated. And here you can see what I mentioned previously, that in Swiss-Prot you can also find the results of programs of sequence analysis data, but these are always verified by and validated by curators. Subcellular location is not only annotated directly using a controlled vocabulary maintained by UniProt, but it also falls under one of the three categories of biological information defined by the gene ontology. As you may know, and we will hear later in more detail, these three categories are biological process, molecular function, and cellular component. And the Go terms that correspond to subcellular location are in the cellular component division of Go and are provided here via the Go annotation tab. But we'll hear a bit more detail about Go later. The next annotation topic we are looking at is post-translational modifications. Over 700 post-translational modifications, or PTMs, are currently annotated in UniProt, and they follow a controlled vocabulary. At the end of our little tour through an entry, we'll take a look at a more plant-specific subject, namely how to find information related to plant pathogen interactions. UniProt is a general database covering all organisms, and data on host pathogen interactions is typically one type of data which is not confined to a single section of the record. Instead, it is distributed across various sections. However, you could search using the tag microbial infection, which may appear in different sections of an entry, such as the function here. We use the tag microbial infection to differentiate the natural characteristics of a protein from those found in a microbial infection context. Additionally, you may search again using Go, and there are various Go terms. In particular, the one which is called response to other organism or its children, for example, defense response to virus. Here we've put a query where we were looking for the tomato proteins involved in defense response to virus. The query is down here, and you can click on it. You will later get the link to the PDF file if you want to use all those links. So you will be able to click on it to see the results. Additionally, you may search using keywords. This is another controlled vocabulary UniProt maintains to help querying for entries, and to search for green plant proteins involved in the process of nodulation. You can use this query. This keyword identifier here corresponds to the nodulation process. And as I showed previously for taxonomy, you don't have to know this identifier. You can use the advanced search where if you type nodulation, this keyword will be suggested. So this is a query for green plant proteins involved in nodulation, but of course you can also make the same query for bacteria. Now I'd like to introduce another concept and another important space on the UniProt website, which is proteomes. A proteome is a set of proteins thought to be expressed by a given organism. Such a set is generally made up of all sequences that correspond to a given genome assembly. Proteomes are available for organisms across the whole tree of life, from viruses to higher eukaryotes. And on our website, they are accessible from this square here, which is called Species Proteomes, and at the URL we're giving here. How many plant reference proteomes are there? First of all, reference proteomes is a subset of all proteomes that are carefully selected, complete sets of proteins believed to be expressed by a specific organism. And these are particularly model organisms or those of interest for research. There are currently almost 300 of them for plants. This is the beginning of the list, in this case, sorted in reverse alphabetical order. And this small section here already includes some interesting organisms such as grapes, ginger, or vanilla. You can click on those proteome identifiers and get to a page like this, where we see that depending on the organism and the extent to which the organism has been studied, there can be more or less reviewed entries in a proteome set. Arabidopsis is a model organism with quite a high number of Swiss-Prot entries. But there are many proteomes for which the vast majority or even all entries are in TrEMBL. The entries that make up a proteome can be downloaded, either all of them or in a set that is gene-centric and that contains one protein sequence per gene. Now let's move on to the biocuration process. Amongst all the fascinating biological subjects, the priority in plant curation is focused on the model organism, Arabidopsis thaliana, and on rice, as well as publications with an agroeconomic impact. From time to time, we launch internal cross-functional projects for specific tasks. And importantly for you to know, we give priority to user requests. So as I'll say in the end, you're very welcome to send update requests and to suggest publications, for example. We also collaborate with other databases and exchange data with them, as they often specialize in data that we don't have to that level of detail. For example, TARE and RIA. Now I'll take you on a short journey with the biocurators, such as Emmanuel here in the room, and we'll see how they go about annotating a new entry. It usually starts with one or more publication. This one is about an enzyme in cannabis sativa. As we saw before, we first want to get the sequence right. The publication hopefully includes some accession numbers or identifiers, and any mentioned gene name, protein name, or accession number will be used to search for a corresponding protein in UniProtKB, but also in other databases. Once a reliable protein sequence has been found, a similarity search is performed using BLAST to find all similar protein sequences in UniProtKB, with the goal to combine all redundant sequences for the same gene into a single entry. In this case, there were 3 candidate protein sequences which were aligned, and then the canonical sequence was constructed. There are small differences in there, which are curated and described as conflicts, or also variants, if they can be recognized as variants. If no UniProtKB entry matches the described protein, a new entry is manually created from scratch, or from the nucleotide sequence databases or other sources, or from short peptides published in the sequence in the publication. This is the corresponding view on the UniProt website. All the protein sequence sources used for the construction of the protein sequences are indicated together with the corresponding conflicts. Approximately 30% of all green plant protein sequences in Swiss-Prot actually required correction in the form of conflicts or similar things that need to be resolved. Now let's look at how our expert curators assemble biological knowledge once they have the sequence. They look at the publication, where knowledge representation is usually targeted to the human reader in form of images, tables, free text, figures. Now the job is to structure the data to make it accessible to computer programs, which can then extract, analyze the data. And it could look like this, for example, in the internal version. This is rather technical, and this is where it's good to have the data accessible to computer programs again, because there can be programs that allow to build a user-friendly presentation of the same data. Now, before we come to the next important step in the biocuration process, I need to finally introduce some details on the gene ontology, which I've already briefly mentioned before. It is a structured, standardized representation of biological knowledge, and it describes concepts that are connected to each other via formally defined relationships. This enables the consistent gene annotation, but also comparison of function against organisms and integration of knowledge across very diverse biological databases. All of this is extremely important to us and the scientific community, which is why Go curation is an important part of the Swiss-Prot curation process. Go is organized in 3 aspects, as you may know: molecular function, and the cellular component, which we've already briefly seen. That was in the context of the subcellular location, and biological process. The Swiss-Prot group, or UniProt, is an important participant in the Go consortium and does a lot of curation that is not only visible in UniProt, but also to all users of Go. For the entry we've been looking at, the curator has compiled a paragraph that summarizes the protein's function in 3 sentences based on the publication here. At the same time, they further abstract this function by assigning gene ontology terms corresponding to the different aspects of the function. The first sentence marked here is, for example, mapped to 2 Go terms related to 2 different biosynthetic processes. If we move on to the second sentence, another biosynthetic process can be identified, which has an associated Go term, and the same for the third sentence. After the function, we move on to the subcellular location, where the curator uses information from a figure and from the text in the publication to describe the subcellular location, used as in-house controlled vocabulary that I mentioned previously, and also by identifying a relevant Go term. Again, the source of the article, the PubMed ID, is provided. Here we see again how the knowledge is structured to become accessible to computer programs. And then again, some of these computer programs can be used to make the data more accessible to humans. In this case, we use a collection of cell illustrations that allow us to depict the protein's localization within the cell. That set of images is called Swiss Biopics. It's also provided by members in our team, but can be used outside of UniProt. And depending on the organism and the type of cell, a different image is automatically selected that allows to best represent the cell and the protein's localization. For example, here it is a plant cell, but bacterial cells, for instance, can look quite differently, or in higher eukaryotes, there are muscle cells, neural cells, and many more. Experimental 3D structures are another important source in biocuration. The entry we looked at previously didn't have an experimental 3D structure, which is why we moved to another protein now to illustrate these aspects of the curation process. The protein is THCA synthase, which is the enzyme controlling psychoactivity of cannabis. 3D structure is an important source for many annotations. Here we see it displayed the structure of the THCA synthase, which is derived from this PDB entry, 3VTE, which is derived from X-ray experiments and allows to visualize, among other aspects, the binding site of the cofactor FAD. Sequence residues involved in the binding of substrates or cofactors, like this FAD, as well as active sites are captured in UniProt in a structured format using KB, which we've already seen in the context of reaction. And all of this is based on a combination of the structure with the underlying publication. If we now want to visualize one of those sites on the structure, we can go to the feature viewer, which is accessible in a different tab. Entry is the main tab, but if you click on feature viewer, you can select if there are different structures. If there's more than one, you can select the one you're interested in, like this one. And then select in the graphical part here, click on the binding site, and we will then see the site in the context of all other annotated features. But not only that, we can also see in the structure viewer the corresponding area highlighted. The viewer jumps to a portion of the structure that will allow us to see the selected part, and it's still manipulable. You can turn it and move it around. It's interactive. In the last part now of today's presentation, we will see a few of the highlights, but also challenges in the annotation of biosynthetic pathways. Reconstructing biochemical pathways presents often a very significant challenge for the curators, as we will see. In this example, we will have a look at the data found in the publication here, focusing on the biosynthetic pathway of the two cannabinoids, CBD and THC, in cannabis sativa. Much of the data on pathways is embedded within figures, graphics, and the enzymes involved in the various reactions are often quite difficult to identify. Understanding the elements of graphical representation often also requires accompanying text. And unfortunately, there's no universal vocabulary for naming small molecules. At UniProt, as mentioned previously, we use KB, the nomenclature from KB, which is standardized at pH 7.3. And more challenges to the biocurator, identifying enzymes catalyzing the reactions in the text can be challenging too. And this is unfortunately not yet handled efficiently by artificial intelligence. And an additional complication is that both references and molecules are often represented by numbers. And that again is a huge difficulty for artificial intelligence systems. And to find additional information on the pathway, we of course refer to the publications cited in the reference list of the article. We also use a tool based on machine learning, which is called LitSuggest, to help identify other pertinent publications. And this tool was actually trained, among others, with input of our curators for the identification of enzymes. Now, after all this effort by the biocurator, all the enzymes involved in our biosynthetic pathway are finally annotated. Enzymes are highlighted in red here, have been recently added to UniProt, along with newly annotated reactions in the RIA database. As you can see, there were previously only acronyms on the left-hand side and numbers. And now we have unique identifiers for proteins in UniProt and the enzyme classification, and also identifiers for reactions in RIA. Looking at such a pathway now, we can understand, for example, that depending on the expression levels of the enzymes THCAS and CBDAS, plants can accumulate different amounts of THC and CBD, which can be quite interesting. Unfortunately, it's currently not possible to retrieve the entire pathway with a single query in UniProtKB. We are in the early stages of developing tools that will hopefully enable the visualization of complete pathways. However, there's an example of what can already be done using UniProt. Let's take a closer look at this small molecule here, hexanoyl-CoA. According to this graph, which the curator has enriched with references to enzymes and reactions, which have all been curated, we can now find, or we can suppose, that this molecule is directly related to two enzymes, AAE1 and OLS. And we'll now verify this in UniProt. So we could look for proteins interacting with hexanoyl-CoA in cannabis. And to do this, we go to the advanced search, select the subsection small molecule from the dropdown that has all the field names, and then enter the molecule name in the search box next to it. You can see that the search engine provides some useful suggestions again, from which we can select the first one. And then we click on search. But actually before that, yeah, we also specify the species, Cannabis sativa, and we are happy to see that the result contains exactly the two expected sequences, OLS and AAE1, which have been curated, which are in Swiss-Prot. If now we wanted to search for entries interacting not only with hexanoyl-CoA, but with molecules that are similar to it, this couldn't be done with the advanced search. To answer this type of more complex questions, we need to use a technology called Sparkle instead, which may look a bit daunting at first for non-experts, but feel free to explore our ready-to-use Sparkle queries. There is a selection available at this URL, which is the Sparkle endpoint, and they can be easily customized to suit your needs. There is actually one query that does exactly what we want to do here, but for molecules similar to dopamine and not to hexanoyl-CoA. So we can take this query, which does what we want to do, but for dopamine. And now we can just take that query and replace all the information that is specific to dopamine and replace it by what we need for hexanoyl-CoA. And we can extract this information from KB. We can get the smiled string and substitute it in the query, and then we'll get our results. Now with this short outlook on Sparkle, which I hope was not too intimidating, we have arrived at the end of today's webinar. I'd like to encourage everyone to contact us if you have questions. Or of course, you can also contact us with bug reports, general feedback, or annotation updates. The contact form is available via the envelope system at the top of the page. And there's also a link to the help center where you can find a wealth of useful documents. And with that, thank you very much for your attention. And we are happy to take questions if you have any.